Next, we're going to talk about how the cells can regulate gene expression after transcription, and we'll call this post-transcriptional regulation. And although this transcriptional control is probably the best studied and possibly the major type of regulation in eukaryotes, the post-transcriptional regulation also plays a very important role, and it's just now being understood just how important that role is. So when we're talking about post-transcriptional regulation, we're talking about things like processing of the mRNA. So we'll talk about how introns can be removed, exons can be spliced together. There's also a cap that gets put on and a poly A tail that gets put on. And it can include downstream things too, such as translation and stability, but we'll primarily talk about those in the next lecture. So approximately 2/3 of human genes that we know of undergoes alternative splicing. And the main benefit of this is that it gives us many more different possible combinations of products from our genes than our genes alone might indicate. And humans produce about several 100,000 different proteins, and we only have maybe 20 to maybe 25,000 genes in our genome. There are known mutations that can affect regulation of splicing, and we'll call these spliceopathies, and they can contribute to many different genetic disorders. And these can be things like myotonic dystrophy, Fragile X syndrome, and Huntington's disease. So when we're talking about processing the mRNA transcripts, we're talking about this. Introns are going to be regions that are in between, and they are not going to be included in the final product. Exons are going to be expressed regions of the gene, and they are going to be included in the final product. Now, which of those exons is going to be included in the final product is up to the individual cells, and that is occurred through this differential splicing. So for example, in one type of cell, you might have all 5 exons, in this case A, B, C, D, E, in the mature mRNA, and this would produce a particular protein product. Now in a different cell, maybe under different conditions, maybe even in the same cell, it can also produce, instead no C here, so it would just be A, B, D, E, and this would give it a different protein product that may have a different function. So here's an example of a particular transcript that can be processed in several different ways. There are going to be 6 exons in this particular transcript, the primary transcript. And you can see here they're not labeled, but in these little reddish brown bars are going to be the regions of the intron. So if you splice it so that you include exons 1, 2, 3, 4, after translation, after processing, this is going to make calcitonin peptide. Now, if you were to take exons 1, 2, 3, skip 4, and take 5 and 6, you can make a different product. In this case, it will be the CGRP peptide after translation and processing. And both of these are going to have very different roles. Here's another example of processing. This processing can also happen so that it is different, not only in different cells, but also in different individuals. So this might depend on things like other hormones in the body. And it can lead to things like sex-specific transcription products. So in this particular case, there's a gene for SXL. Okay, this is going to regulate splicing in this particular product. So in this case, this protein is going to have female-specific splicing, okay, and you're going to make a particular product that is going to be specific for females. In males, because they don't have this SXL gene, or at least don't do the transcription of this particular gene, you're going to have male-specific splicing, and you're going to get a different product, leading, in this case, DSXM protein, instead of the DSXF protein. And this is going to be important for male differentiation.